Good day, this is the Professor, CBMD PhD. Today is Monday, October 23rd, 2023. It is 5:49 and 3 seconds, AM Eastern Standard Time. And today's research, molecular phage. So let's jump to it here. Molecular phage in the eye, determined. Date October 19th, 2023. Source, Cell Press. A team of researchers have mapped almost 6,000 proteins from different cell types within the eye by analyzing tiny drops of eye fluid that are routinely removed during surgery. The researchers used an artificial intelligence model to create a proteomic plot from this data that can predict a healthy person's age based on their protein profile. The plot revealed that diseases such as diabetic retinopathy and uveitis causes accelerated aging within specific cell types. And surprisingly, the researchers also detected proteins associated with Parkinson's disease within eye fluid, which they say could offer a pathway to earlier Parkinson's diagnosis. What is amazing about the eye is we can look inside and see diseases happening in real time. The primary focus was to connect those anatomical changes to what is happening at the molecular level inside the eyes of the patients. The eye is a difficult organ to sample in living patients because, like the brain, it is non-regenerative and taking a tissue biopsy would cause irreparable damage. An alternative method is to use liquid biopsies, samples of fluid taken from near the cells or tissues of interest, low liquid biopsies, to provide a snapshot of what proteins are present in the region of interest. They have thus far been limited in their ability to measure large numbers of proteins within the small volumes of fluid, and they are also unable to provide information on which cells produce which proteins, which is important for diagnosing and treating diseases. And to map protein production by different types of cells within the eye, the team used a high-resolution method to characterize proteins in 120 liquid biopsies taken from the aqueous or vitreous humor of patients undergoing eye surgery. Altogether, they identified 5,953 proteins, 10 times the number of proteins previously characterized in similar studies using a software tool they created called Tempo. The researchers were able to trace each protein back to specific cell types and to investigate the relationship between disease and molecular aging. The researchers built an artificial intelligence machine learning model that can predict the molecular age of the eye based on a subset of 26 proteins. The model was able to accurately predict the age of healthy eyes, but showed that diseases were associated with significant molecular aging. For diabetic retinopathy, the degree of aging increased with disease progression, and this aging was accelerated by as much as 30 years for individuals with severe proliferative diabetic retinopathy. The signs of aging were sometimes observable before the patient displayed clinical symptoms of the underlying disease and lingered in patients who had been successfully treated. The researchers also detected several proteins that are associated with Parkinson's disease. These proteins are usually identified post-mortem, and current diagnostic methods aren't capable of testing for them, which is one reason Parkinson's diagnoses are so difficult. The screening for these markers in eye fluid can enable earlier diagnosis of Parkinson's disease and later therapeutic monitoring. The scientific investigators and microbiologists say that these results suggest that aging may be organ or even cell-specific, which can yield advances in precision medicine and clinical trial design. These findings demonstrate that how organs are aging at different rates. The use of targeted anti-aging drugs could be the next step in preventative precision medicine. If we are going to use molecular therapies, we should be characterizing the molecules in the patients. They think that reclassifying patients based on their molecular patterns and which cells are being affected can really improve clinical trials, drug selection, and drug outcomes. Next, the researchers plan to characterize samples from a larger number of patients in a broader range of eye diseases They also say that their method can be used to characterize other difficult to sample tissues. As for example, liquid biopsies of cerebral spinal fluid could be used to study or diagnose the brain. Synovial fluid could be used to study joints, and urine could be used to study the kidneys. And this is a professor of microbiology, bioinformatics, pathophysiology, and teacher of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.